Purine and pyrimidine metabolism. The main questions of the lecture: overview, nucleotides metabolism, pyrimidine metabolism, and purine metabolism. Nucleotides are needed for DNA and RNA synthesis, DNA replication and transcription, and for energy transfer, and they're used as signal molecules. Nucleoside triphosphate, ATP, and GDP provide energy for reactions that would otherwise be extremely unfavorable in the cell. Ribose biphosphate for nucleotide synthesis is derived from the hexose monophosphate shunt and is activated by the addition of pyrophosphate from ATP, forming phosphoribosyl pyrophosphate using PRPP synthase. Cells nucleoside synthesis in two ways: the de novo synthesis and salvage pathway. And de novo synthesis means new, which occurs predominantly in the liver. Purines and pyrimidines are synthesized from smaller precursors, and PRPP as is added to the pathway at some point. In the salvage pathway, performed purine and pyrimidine bases can be converted into nucleotides by salvage enzymes, distinct from those of the de novo synthesis. Purine and pyrimidine bases for salvage enzymes may arise from synthesis in the liver and transport to other tissues, digestion of endogenous nucleic acids. In many cells, the capacity of de novo synthesis is supply purines and pyrimidines insufficient, and the salvage pathway is essential for adequate nucleotide synthesis. In Lesch-Nicken disease, an enzyme for purine salvage hypoxanthine guanine phosphoribosyl pyrophosphate transferase is absent or deficient. People with this disease deficiency have CNS deterioration, mental retardation, and spastic cerebral palsy associated with compulsive cell mutilation. Cells in the basal ganglia of the brain normally have very high activity of this enzyme. Patients also all have hyperuricemia because purines cannot be salvaged, causing gout. Pyrimidine synthesis. Pyrimidines are synthesized de novo and in the cytoplasm from aspartate, CO2, and glutamine. Synthesis involves a cytoplasmic carbamyl phosphate synthase that differs from the mitochondrial enzyme, which the same name used in the urea cycle. The primary end product of pyrimidine synthesis is UMP. In the conversion of UMP to DTMP, important enzymes are ribonucleotide reductase, thymidylate synthase, and dihydrofolate reductase, all targets of antineoplasmic drug. Ribonucleotide reductase reduces all NDPs and DNDPs for DNA synthesis. It inhibits by hydroxyurea. Thymidylate methylates DUMP and DTMP, requires THF. 5-fluorouracil inhibits this activity of this enzyme. The hydrofolate reductase converts DHF to THF. Without DHF reductase, thymidylate synthesis will eventually stop. This enzyme inhibited by methotrexate, trimethyltryptamine, and pyrimetamine. Ribonucleotide reductase is required for the formation of the deoxyribonucleotides for DNA synthesis. All 4 nucleotide substrates must be diphosphates. DTP and DATP strongly inhibit ribonucleotide reductase. Hydroxyurea, an anti-cancer drug, blocks DNA synthesis indirectly by inhibiting ribonucleotide reductase. Pyrimidines may be completely catabolized to form ammonia as a produce or recycled by pyrimidine salvage enzyme. Purine metabolism. Purines are synthesized de novo beginning with PRPP, the most important enzyme in PRPP aminotransferase, which catalyzes the first and rate-limiting reaction of the pathway. It is inhibited by their three purine nucleotide and products AMP, GMP, and IMP. The drug celeperenol used for gout and 6-mercaptopurine also inhibit PRPP aminotransferase. These drugs are purine analogs which must be converted to their respective nucleotides by HGPRT within cells. Their amino acids, glycine, aspartate, and glutamic acid, are used in purine synthesis. The tetrahydrofolate is required for synthesis of all the purines. Adenosine monophosphate is the precursor for AMP and GMP. Excess purine nucleotides, or those released from DNA and RNA by nucleases, are catabolized first to nucleosides, loss of P inorganic, and then to free purine bases, release or ribose or deoxyribose. Excess nucleoside monophosphates may accumulate when RNA is normally digested by nucleases. Dying cells release DNA and RNA, which is digested by nucleases. The concentration of free inorganic phosphate decreases, as it may in galactosemia, hereditary fructose intolerance, and glucose 6-phosphatase deficiency. Salvage enzymes recycle normally about 90% of these purines, and 10% are converted to uric acid and excreted in urine. When purine catabolism is increased significantly, a person is at risk for developing hyperuricemia and potentially gout. Purine catabolism to uric acid and salvage of the purine bases hypoxanthine and guanine are shown below on this slide.